Okay, hello to all of you. Let's discuss chapter number 9. Chapter number 9 is about cellular respiration. So let me go into the slideshow mode. So, cellular respiration, first of all, we need to understand what this process of cellular respiration means. So, cellular respiration is a process in which actually the energy is extracted from the food we have eaten. So, this is a process in which the energy is extracted from the food we have eaten. So, as we have already discussed in chapter number 8, we have already talked about what is energy, why do we need energy. So these things we have already discussed. So basically energy flows into an ecosystem as sunlight and leaves as heat. So basically the main source of energy in our ecosystem is sun. So the main energy is coming from sun, and the plants, they have the photosynthetic pigments in them, and because of that they can undergo the process of photosynthesis, and they are able to convert the light energy into the chemical energy. And then this chemical energy is basically these these plants, they can convert light energy to chemical energy, which is in the form of organic molecules, and the animals, which we also belong to the animal kingdom. So we eat either directly plants, or we eat animals which were fed on the plants. So we are directly or indirectly dependent on the plants, and the plants are actually dependent on the sun. So the main source of energy in our ecosystem is coming from the sun. So basically, there is a cycle going on, photosynthesis and the cellular respiration cycle going on. So photosynthesis is a process which converts light energy into the chemical energy in the form of organic molecules. Also, another important thing about photosynthesis is another product of photosynthesis is oxygen. So these organic molecules, they are eaten by the animals, and then in the cells of the animals, by the process of cellular respiration, the energy is extracted from these organic molecules. And in this process of cellular respiration, in which the energy is extracted from the food we have eaten, oxygen is also required, because oxygen, in this process, the oxygen acts as a final electron acceptor. So oxygen acts as a final electron acceptor, and the energy is produced in the form of ATP. And the byproducts of cellular respiration are carbon dioxide and water, and they are taken by the plants or the photosynthetic organisms. So they are taken by plants or the photosynthetic organisms, and they are used in the process of photosynthesis, and they convert these carbon dioxide and water into organic molecules and oxygen. So this is a cycle going on. And we are going to talk about basically the process of cellular respiration. Important thing to know about the process of cellular respiration: it is a catabolic process, and in this energy is released, and mostly the energy is in the form of electrons. So we are going to discuss that, and you will see that how the electrons transfer and how the energy is created. So before we move forward, we need to know about some of the terms. What is fermentation, what is aerobic respiration, and what is anaerobic respiration. So aerobic respiration is a process in which energy is released, in which oxygen acts as the final electron acceptor. And anaerobic respiration is a process in which energy is released, but the only difference between aerobic and anaerobic respiration is the final electron acceptor in anaerobic respiration is not oxygen, but it is something else. It can be some protons, it can be nitrates, sulfates, but it is not oxygen. So third process is fermentation. So fermentation is different from anaerobic respiration. It is different from anaerobic respiration because in the fermentation is an anaerobic process. In this process, an oxygen is not involved. So in the process of fermentation, oxygen is not involved. So the degradation of sugar takes place, and it's a partial degradation of sugar which takes place in the fermentation, and it takes place in the absence of oxygen. So there's a difference between, I repeat, there's a difference between anaerobic respiration and fermentation. In the anaerobic respiration, the process is quite same as aerobic respiration, but the final electron acceptor is different. It is not oxygen. It is something else than oxygen. So that's why it's called anaerobic respiration. But fermentation is a anaerobic process, which means it's a process which takes place in the absence of oxygen. It takes place in the absence of oxygen, and it is a partial degradation of the sugars. So cellular respiration basically involves both aerobic and anaerobic respiration, but it is most often used for aerobic respiration. And this is the equation. So usually in our food, we not only eat carbohydrates, we also eat lipids, we also eat proteins. But when we talk about the cellular respiration process or the aerobic respiration process, we usually talk in terms of glucose. So this is glucose, and it reacts with oxygen, and it produces carbon dioxide and water and ATP. And so basically, this is a process, cellular respiration is a process in which energy is produced. And we're going to see that the energy is basically in the form of electrons. And in this process, the transfer of electrons will take place so that the electrons are either lost or they are gained. And the loss of electrons is called oxidation, and the gain of electrons is called reduction. So these kind of reactions in which there is a gain or the loss of electrons takes place, these kind of reactions are called redox reactions. So oxidation is the loss of electrons and reduction is the gain of electrons. So here is basically a simple example of sodium chloride. So sodium chloride is a ionic compound, as you know, we have already talked about. In this, so when sodium and chlorine, these ions, they react, when sodium and chlorine ions, when they form bond with each other, they form the compound called sodium chloride. And in this, sodium basically loses electrons and chlorine gains electrons. So sodium is oxidized and chlorine is reduced. So sodium is oxidized and chlorine is reduced. So the electron donors are called reducing agents because electron donors are the ones which are making other molecule or other ion reduce. So the electron donors are reducing agents, and the electron receptors, any compound or any atom or any ion which is receiving the electrons, they are the oxidizing agents. And this is example. So we just talked about sodium and chlorine, which is an ionic compound. Here is an example of methane reacting with oxygen, forming carbon dioxide and water. So in this reaction, which involves methane and carbon dioxide, and these compounds, they have covalent bonds in them. So let's see how these atoms, they're getting reduced and oxidized. So if you look in methane, this is carbon and it is bonded with 4 hydrogens. So this is carbon which is bonded with 4 hydrogens. Carbon, when it is bonding with 4 hydrogens, the carbon is sharing the electron. This carbon is sharing 1 electron with this hydrogen, it's sharing 1 electron with this hydrogen, it's sharing 1 electron with this hydrogen, it's sharing this electron with this hydrogen. So basically, between this carbon and hydrogen, there's a 1 pair of electrons which is being shared. Between this carbon and hydrogen, 1 pair of electrons being shared. So with all these hydrogens, carbon is sharing 1 pair of electrons. So that's happening, and there's a single bond between the carbons and the hydrogens. So here, because there is not much difference between the electronegativity, there's not much difference in the electronegativity of carbon and hydrogen, the electron pair is shared equally between carbon and hydrogen. So when methane reacts with oxygen, so in oxygen, these are 2 oxygens which are forming double bond with each other, 2 oxygens forming double bond with each other, and there is these 2 oxygens, they are sharing these 2 oxygens, they are sharing 2 pairs of electrons with each other, and these because here the partners are both oxygens, because the partners here are both oxygens, these electron pairs are shared equally between these 2 oxygens. So now when the reaction takes place between methane and oxygen, it results in the formation of carbon dioxide. So in the carbon dioxide, you can see here, so there's a double bond between this carbon and oxygen, there's another double bond between this carbon and oxygen. So you can see here, this double bond is between carbon and oxygen. So the electrons are being shared between oxygen and carbon. So the partners are different here. So if we compare the electronegativity of carbon and oxygen, oxygen is more electronegative. Oxygen is more electronegative, and as a result of that, what happens is the electrons are pulled towards oxygen. Because oxygen, if you compare oxygen and carbon, oxygen is more electronegative, so the electrons are being pulled towards the oxygen. So in this case, what happens is this oxygen is getting reduced because the electrons, they are getting more pulled towards the oxygen. So the oxygen in the carbon dioxide, the oxygen in the carbon dioxide is getting reduced. Okay, and similarly, if we look at the carbon in the methane, if we look at the carbon in the methane, the electrons, they were shared equally between carbon and hydrogen. But now if we look in water which is formed, the oxygen, we will come back to the same concept, that carbon and hydrogen, they are sharing electrons equally with each other, right? But what is happening here is this oxygen is getting electrons more towards itself. So in this case, in carbon dioxide, these electrons are moving towards oxygen and they're moving far away from the carbon. They're moving far away from the carbon. So it means that this carbon, which is in methane here, now it becomes carbon dioxide, it gets oxidized because the electrons move away from them and the oxygen gets reduced. Okay. So the carbon, when it converts, when the carbon gets converted into when it gets converted from methane to carbon dioxide, this carbon gets oxidized because the electrons move away from the carbon. But here in the case of, if you look in this oxygen, if you look here, between these two oxygens, they are sharing the electrons equally between each other. They're sharing electrons equally between each other. But now when this oxygen gets converted into water, look here, this is oxygen, these are hydrogens. So sorry, we talk in terms of oxygen, not here. I think I said something wrong before. We talk in terms of oxygen in water. So oxygen here, the electrons are shared equally. Between these two oxygens. But now when it forms water, these electrons, they move more towards the oxygen, because if we compare hydrogen and oxygen is more electronegative. So the electrons move towards the oxygen. And as a result of that, here oxygen is getting reduced, because here the electrons were shared equally, but now the electrons are moving more towards the oxygen. So here oxygen is getting reduced. So this is what basically oxidation and reduction is. So in this process of cellular respiration, it's a process in which energy is produced. As I said, energy is in the form of electrons. But remember that electrons are subatomic particles, right? Remember from previous chapters, we have electrons, we have protons, we have neutrons. So electrons never run alone. They never run alone as the subatomic particles. So electrons in our food, they are in the form of hydrogens. So electrons run in the form of hydrogen atoms. So electrons, they are present in the form of hydrogen atoms. So hydrogen atoms, they have one electron and one proton. They do not have any neutrons. So one electron, and so hydrogen atom has one electron and one proton. And the electrons, they run in the form of hydrogen atoms. So this is glucose, this is oxygen, and the products are carbon dioxide and water and energy. And in our cells, I would like to tell you, today I have posted some of exercises for you guys. and in there are many cartoons which show that we have two electron carriers in our cells. One is NAD plus, and it is basically a derivative of a vitamin. So we have two electron carriers, NAD plus and FAD. So they are coenzymes, and they are the ones which carry the electrons. So these electrons, they come from the food we have eaten, and these are the ones, these electron carriers are the ones which take these electrons to the destination. And what is the destination of the electrons? The destination of the electrons is the destination of the electrons is the inner membrane of mitochondria. So we have already talked about the structure of mitochondria. The inner membrane of mitochondria is all invaginated, and it's called cristae. And in this inner membrane of mitochondria are embedded some protein complexes. And these electrons, which are high energy electrons, which are coming from the food we have eaten, they are going to flow through these electrons, and we will study what happens. It's a process called oxidative phosphorylation. But these electron carriers, NAD plus, and there's another electron carrier called FAD, they are the electron carriers. They are the ones which carry the electrons to the inner membrane of mitochondria. So this is the NAD plus. Okay, so this is nicotinamide adenine dinucleotide. That's the full form of NAD plus. Yeah, so important thing to know about NAD plus is it has a positive charge. So it means that, so this is the structure of NAD plus. Okay, it has positive charge on it, which means it has one electron less. It's charged. So so here you can see these are two hydrogen atoms which are being shown here, and they are coming from the food we have eaten. So two hydrogen atoms means two electrons and two protons. So two hydrogen atoms means two electrons and two protons, and one of the electron is used to neutralize this charge. Okay, so there are two electrons. From these two electrons, one of the electron is used to neutralize this charge, and it gets converted from NAD plus to NAD, and then one more electron and one more proton is going to combine with NAD, and it's going to convert into NADH, and there will be release of one proton. I hope you guys are understanding. So these two hydrogen atoms, they're coming from the food we have eaten, and every hydrogen atom has one electron and one proton. Okay, so one electron, so two hydrogen atoms, have two electrons and two protons. NAD plus has a charge on it, which means it has one electron less. So one electron will be used to neutralize this charge, and this NAD plus will get converted into NAD, and then there is one electron here and two protons. One electron and one proton. One electron, one proton means one hydrogen atom will be combining with NAD, and it will be converted into NADH. As there were two protons, one proton will be released. And NAD plus, when it gets converted into NADH, it gets reduced because it is now gaining electrons. So it gets reduced, and NADH, it passes the electrons to the electron transport chain, and electron transport chain is present in the inner membrane of mitochondria, and at the end of the electron transport chain there is oxygen, and this oxygen, it gains these electrons which will be flowing through the different protein complexes which are present in the inner membrane of mitochondria. And these oxygen, when it gains the electrons, this oxygen gets reduced, and it converts into water. So here is basically shown the difference between combustion and cellular respiration. In cellular respiration, we are going to talk about all the steps of cellular respiration, and you will see that the energy comes out in different steps. Okay, so it's a process in which energy does not come in one step; it comes in small steps, and we will be talking about all these steps. So basically, cellular respiration process involves different stages. First of all is glycolysis, and then it is citric acid cycle, and then last is oxidative phosphorylation. So glycolysis is the first step of cellular respiration in which glucose is broken into two molecules of pyruvate, and then there is a connecting step in between glycolysis and citric acid cycle. We are going to talk about that, and then citric acid cycle goes through some reactions, and then the last step is oxidative phosphorylation, which takes place in the inner membrane of mitochondria. So this is glycolysis. So important thing to know about the first step in cellular respiration is glycolysis. It takes place in the cytoplasm. It takes place in the absence of oxygen, and in glycolysis, glucose is converted into pyruvate, and in this only two molecules of ATP are produced, and they are produced by a process called substrate level phosphorylation. So in this whole process of cellular respiration, there are two kinds of processes involved by which ATP is produced. One is called substrate level phosphorylation, and second is called oxidative phosphorylation. So in the first step in glycolysis, glucose is converted into pyruvate and ATP is produced. It takes place in cytoplasm. It takes place in the absence of oxygen, and then pyruvate is converted into acetyl coenzyme A. Pyruvate is converted into acetyl coenzyme A, and this is called the conversion step, and this takes place in the mitochondrial matrix. And then this acetyl coenzyme A goes into the citric acid cycle, and this citric acid cycle also takes place in the mitochondrial matrix, and in this process also ATP is produced, and this ATP is produced by the process substrate level phosphorylation. And at all these steps, whether it's glycolysis or pyruvate oxidation or citric acid cycle, in all these steps the electrons are stripped from the food, and they're taken by the electron carriers. There are two electron carriers involved, NAD plus and FAD, and they finally take these electrons to the inner membrane of mitochondria, and these electrons, they flow through the different protein complexes which are embedded in the inner membrane of mitochondria, and these electrons, which are carried by the electron carriers, NADH and the electron carriers are NAD plus and FAD, and they get reduced to NADH and FADH2, and these high energy electrons, they flow through these proteins, and eventually the energy is produced by a process of oxidative phosphorylation, which involves electron transport chain and chemiosmosis, and I'm going to discuss that. And lot of energy is produced, in the last step, and this production of ATP is produced by the process of oxidative phosphorylation. So in this step of glycolysis and the Krebs cycle, the energy is produced by substrate level phosphorylation, and in the last step the energy is produced by the process of oxidative phosphorylation. And the process of oxidative phosphorylation, it actually produces around 90% of the ATP, and only a small amount of ATP is produced by the process of substrate level phosphorylation. So this basically shows you what the process of substrate level phosphorylation is. The process of substrate level phosphorylation involves the conversion of ADP into ATP, and this process involves the enzyme. And in this process, when ADP is converted into ATP, so in this process, when ADP is converted into ATP, the source is some intermediate metabolite. Okay, so you can see this is an intermediate metabolite. It has a phosphate group. So this phosphate group is given to ADP. This phosphate group is given to ADP, and it is converted into ATP. So from adenosine diphosphate, it's converted into adenosine triphosphate, and it takes place with the help of the enzyme. So this process is called substrate level phosphorylation. So in the whole process of cellular respiration, around 32 molecules of ATP they are produced. So let's talk about glycolysis. So glycolysis takes place in cytoplasm. That's important thing to know. Please go over the study guide, and all these important things are written in the study guide. So glycolysis takes place in cytoplasm. It takes place in the absence of oxygen. It can take place. I mean, oxygen is not required for this process. So it's not. So glycolysis occurs whether or not oxygen is present. And glycolysis involves two stages. One is energy investment phase, another is energy payoff phase. So in the first phase, energy is invested. In the second phase, energy is harvested. So this basically overall shows you there is a, in the glycolysis, first there is energy investment phase in which 2 molecules of ATP they are used, and then in the payoff phase there are 4 molecules of ATP which are formed. So the net overall outcome of glycolysis is 2 molecules of ATP, and in this process 2 molecules of NAD plus they are also reduced. Okay, and from 2 molecules of NAD plus 2 molecules of NADH they are produced. So the overall outcome of glycolysis is 1 molecule of glucose is converted into 2 molecules of pyruvate. 1 molecule of glucose is converted into 2 molecules of pyruvate. In this whole process of glycolysis, 2 molecules of ATP are produced, and also so the net outcome of ATP is 2 molecules, and also 2 molecules of NAD plus they are converted into 2 molecules of NADH. So NAD plus is reduced to 2 molecules of NAD plus, they are reduced to 2 molecules of NADH. So this is overall glycolysis. So now let's go and talk about the steps of glycolysis. So for you guys, you do not need to know the enzymes, but you need to know what is happening in every step. So in glycolysis, glucose is converted into glucose 6-phosphate. So when glucose is converted into glucose 6-phosphate, there is a requirement of a phosphate group. And from where this phosphate group comes? This phosphate group comes from ATP. ATP gets converted into ADP. So glucose gets converted into glucose 6-phosphate, and the source of phosphate is ATP. ATP gets converted into ADP. Okay. So here the investment of energy is required here. Okay. So this is the first step where the input of energy is required. Then glucose 6-phosphate is converted into fructose 6-phosphate. So you know glucose and fructose, they are isomers, and then fructose 6-phosphate is converted into fructose 1,6-bisphosphate. So here again there is a additional input of ATP is required here, okay, because another phosphate group is required. So fructose 6-phosphate gets converted into fructose 1,6-bisphosphate, and then this fructose 1,6-bisphosphate gets cleaved. It gets cleaved into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, it gets cleaved and it gets broken into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So these are also isomers. They are interconvertible, and after this step, basically dihydroxyacetone phosphate can get converted into glyceraldehyde 3-phosphate. So dihydroxyacetone phosphate gets converted into glyceraldehyde 3-phosphate. So after this step, there are 2 molecules of glyceraldehyde 3-phosphate. So everything, whatever is produced, will be multiplied by 2. So glyceraldehyde 3-phosphate. So remember that there are 2 molecules of glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate gets converted into 1,3-bisphosphoglycerate. In this step, what is happening is this glyceraldehyde 3-phosphate is getting oxidized. So glyceraldehyde 3-phosphate is getting oxidized, which means that it is from glyceraldehyde 3-phosphate, electrons are being lost. So from glyceraldehyde 3-phosphate is getting oxidized. It's getting oxidized, and it is losing electrons. And who is taking these electrons? NAD plus. Who is NAD plus? NAD plus is the electron carrier. So NAD plus is going to accept the electrons which are coming from here. It's going to accept the electrons which are coming from here, and it is going to get reduced. So NAD plus gets converted into NADH. It's getting converted into NADH. So 1,3-bisphosphoglycerate then gets converted into 3-phosphoglycerate. So here there is a loss of phosphate group. So where this phosphate group is now going? This phosphate is going to ADP, and it is going to convert it into ATP, adenosine triphosphate. So this is an example of substrate level phosphorylation, because here ADP is getting converted into ATP, and who is the source of phosphate? The source of phosphate is this compound, which is 1,3-bisphosphoglycerate. And 3-phosphoglycerate is formed here because this compound is losing one phosphate and it is giving to ADP, it's getting converted into ATP. Then this 3-phosphoglycerate gets converted into 2-phosphoglycerate, and then this 2-phosphoglycerate gets converted into phosphoenol pyruvate. Okay, it gets converted into phosphoenol pyruvate, and then this phosphoenol pyruvate eventually gets converted into pyruvate. Phosphoenol pyruvate, it gets converted into pyruvate. Here again there is a loss of phosphate group, and this phosphate group is given to ADP and it gets converted into ATP. So again, this is an example of substrate level phosphorylation. So after this step, whatever is happening is getting multiplied by 2. So 2 molecules of NAD plus, 2 molecules of ATP being produced here, 2 molecules of ATP being produced here. So overall in the second phase, which is an energy harvesting phase, 4 molecules of ATP are produced, but because there were 2 molecules of ATP used in the energy investment phase, the net gain of ATP is 2 molecules. Okay, so there's a net gain of 2 molecules of ATP in glycolysis, and also there are 2 molecules of NAD plus, they get reduced to 2 molecules of NADH. Okay, so now from 1 molecule of glucose, there are 2 molecules of pyruvate formed. Glucose has 6 carbons and pyruvate has 3 carbons. So after this, what happens? After glycolysis, there is a conversion step. So I can show you here the conversion step. So this is pyruvate. It has 3 carbons. You can see here they have 3 carbons. So in the conversion step, first of all, there are 3 important things which takes place in the conversion step. And in this conversion step, there is no ATP produced. And this conversion step takes place in the mitochondrial matrix. So this step takes place in the mitochondrial matrix. And there is loss of carbon dioxide. So this conversion step is the first step where the first molecule of carbon dioxide is produced. And this is that carbon dioxide which we exhale. Okay, this is that carbon dioxide which we exhale. Number 2, when there is a loss of carbon dioxide, which means 1 carbon is lost. So this is pyruvate. It is left with 2 carbons. So the group now which is formed is acetyl, and it combines with coenzyme A. It combines with coenzyme A and it results in the formation of acetyl coenzyme A. Also very important thing which is taking place here is there is also the oxidation of pyruvate taking place. So pyruvate is losing some electrons. So there is an oxidation of pyruvate taking place, and when the oxidation of pyruvate is taking place, which means that it is losing electrons. So where are those electrons going? Those electrons are accepted by NAD plus. Those electrons are accepted by NAD plus, and NAD plus is converted into NADH. So these are the 3 important things taking place during this conversion step. Number 1, production of carbon dioxide, combining of coenzyme and the production of acetyl coenzyme A, and also the production of NADH. No ATP is produced here. So after acetyl coenzyme A is formed, it enters into a cycle called citric acid cycle. So I'm going to go over here and explain you from here actually. So the conversion step as well as the citric acid cycle, they takes place in the mitochondrial matrix. Again I'm saying follow my study guide. This chapter will become easy for you, and I have also posted many activities. Please do that at home. They will really help you. So here this is the acetyl coenzyme A which is coming from the conversion step, and there is an oxaloacetate. It's a compound with 4 carbons in it. Okay, it is already there in our cells. So oxaloacetate is a compound with 4 carbons in it. It is already there in the cells. It combines with acetyl coenzyme A. Acetyl coenzyme A has 2 carbons, and they together form a compound called citrate. Citrate has 6 carbons in it, and then this citrate gets converted into isocitrate. Then this isocitrate gets oxidized. Okay, it's losing electrons. If it is losing electrons, who is taking these electrons? These electrons are taken by NAD plus, and NAD plus gets converted into NADH. Okay, and another important thing which is taking place here is this isocitrate. This isocitrate is getting oxidized, and these electrons are taken by NAD plus, and NAD plus is getting reduced to NADH. These are redox reactions. Oxidation and reduction always take place at the same time. One compound is going to be oxidized and the other compound is going to be reduced. So here isocitrate is getting oxidized and NAD plus is getting reduced. Okay. And another important thing which is taking place here is loss of carbon dioxide. So here isocitrate is losing one carbon in the form of carbon dioxide. So isocitrate is getting converted into a compound called alpha ketoglutarate. Alpha ketoglutarate has 5 carbons in it because it has already lost one carbon as carbon dioxide. Now in the next step, alpha ketoglutarate also gets oxidized and the electrons, they are taken by NAD plus, and NAD plus gets reduced to NADH. Okay. Alpha ketoglutarate gets oxidized, it gets oxidized, and NAD plus gets reduced. Redox reaction, again there is a loss of one carbon dioxide. Okay. So alpha ketoglutarate, which has 5 carbons, is losing now one carbon, and now a compound is going to be formed with 4 carbons. And also there is also addition of coenzyme A. So the compound which is formed here is called succinyl coenzyme A. Succinyl coenzyme A has 4 carbons. The succinyl coenzyme A then converts into succinate. So the conversion of succinyl coenzyme A to succinate, this is a very important reaction because here substrate level phosphorylation is taking place and ATP is produced. So this is the only step in Krebs cycle where ATP is produced. Okay. And then this succinate gets converted into fumarate, and when succinate gets converted into fumarate, again succinate gets oxidized, and here the electron carrier is different. The electron carrier is FAD, and FAD gets converted into FADH2. Okay. And then this fumarate, so succinate gets oxidized, FAD gets reduced, succinate gets converted into fumarate, then this fumarate is converted into malate, and then this malate gets converted into oxaloacetate. Now when malate gets converted into oxaloacetate, malate is again getting oxidized and NAD plus is getting reduced and it's getting converted into NADH. So so malate gets converted into oxaloacetate. So we started with oxaloacetate, we are ending up with oxaloacetate. That's why it's called a cycle. So we start with oxaloacetate, we end up with oxaloacetate. So acetyl coenzyme A, which is coming from the food we have eaten, has 2 carbons. It combines with oxaloacetate and it forms a citrate, and the cycle goes on. But important thing to know here is the acetyl coenzyme A. So we started with 1 molecule of glucose. 1 molecule of glucose forms 2 molecules of pyruvate. 2 molecules of pyruvate forms 2 molecules of acetyl coenzyme A. So only 1 molecule of acetyl coenzyme A. So only 1 molecule of acetyl coenzyme A enters into this cycle. So only 1 molecule of acetyl coenzyme A enters into a cycle. So only 1 molecule of acetyl coenzyme A enters into the cycle. So 1 by 1 acetyl coenzyme A enters into the cycle. So how many molecules of acetyl coenzyme A are there? There are 2. So which means that this cycle operates 2 times. So what's the output of one cycle of one Krebs cycle? 1 molecule of NADH here, second here, third here. 3 molecules of NADH are produced in this cycle. 1 molecule of FADH2 are produced. 1 molecule of FADH2 is produced, and 1 molecule of ATP is produced. Okay. And how many carbon dioxides are produced here? 2. 1 carbon dioxide was produced in the conversion step, and 2 molecules of carbon dioxide are produced here. Okay. So if you have to tell the overall output of Krebs cycle, everything needs to be multiplied by 2. So 6 molecules of NADH, 2 molecules of FADH2, 2 molecules of ATP, and 4 molecules of carbon dioxide. This is the total output of your Krebs cycle. Okay. So till we reach here, the food is totally oxidized. Okay. Now all the energy which has been produced till now is produced in the form of 4 molecules of ATP. 2 molecules of ATP were produced in glycolysis and 2 molecules of ATP are produced in the Krebs cycle. So only 4 molecules of ATP they have been produced till now, and they're produced by substrate level phosphorylation. So where is the all other energy? All other energy is in the form of the high energy electrons, and those high energy electrons, they are being carried by two very important electron carriers. One is NAD plus and another is FAD. So NAD plus gets reduced to NAD plus gets reduced to NADH, and FAD gets reduced to FADH2. So where these molecules, where these electron carriers take these electrons, they take these electrons to the inner membrane of mitochondria. Let me explain you from here. Okay, so this is the inner membrane of mitochondria. So all the invaginations you see here, these are the inner membrane of mitochondria, and in these inner membrane of mitochondria is embedded the electron transport chain. So what is electron transport chain? Electron transport chain is this. So this is electron transport chain is basically some protein complexes. These electron transport chain is some protein complexes which are embedded in the inner membrane of mitochondria. So these are different protein complexes which are, there are 4 protein complexes, and these protein complexes, they have some metals in them, and they're called cytochromes. So electrons are passed through number of proteins including cytochromes, each with an iron atom. So there are different kinds of you can see there are different kinds of protein complexes here, and the protein complexes with iron in them, they're called cytochromes. And there are other there are some proteins with copper in them, some have iron and sulfur complexes in them. So these are 4 protein complexes which are embedded in the inner membrane of mitochondria. And the electrons, the high energy electrons which are coming from the food we have eaten, they are being carried by the electron carriers, which are NAD plus and FAD. They carry the electrons, they themselves get reduced, and they bring the electrons at this place in the inner membrane of mitochondria where there is electron transport chain, and they deliver the electrons here. So the NADH delivers the electrons to the first complex, FADH2 delivers the electrons to the second complex, NADH gets converted into NAD plus, FADH2, when it delivers the electrons, it gets converted into FAD. So these electron carriers, when they bring the electrons, they get reduced. When they deliver these electrons to the inner membrane of mitochondria, they get oxidized again, and they go back and they continue their work, and their work or the job is to keep on bringing the electrons, because we keep on eating the food. The food has the high energy electrons. So these electrons, they will be flowing through these proteins, and they are flowing through these proteins because at the end of the electron transport chain, there is oxygen, and oxygen is very electronegative. Oxygen is very electronegative, and all these electrons, they are being pulled through these proteins because at the end of the electron transport chain there is oxygen, and this oxygen, when it receives these electrons, this energy has been released on the way, and I'm going to show you what happens with that energy, what is produced on the way. And these low energy electrons, they're accepted by oxygen, and oxygen gets reduced and it forms water. So water is one of the byproduct of cellular respiration, and now you understand how this water is formed. So now let me explain you from here actually. Now, as I said, NADH delivers the electrons to the first complex, FADH2 delivers the electrons to the second complex, which is here. So these electrons, they're delivered to this is the inner membrane of mitochondria, This is the inner membrane of mitochondria, and in this inner membrane of mitochondria are embedded these protein complexes, and these protein complex and these electrons, they flow through these protein complexes, they flow through these protein complexes, and the energy is released, and that energy is used to pump the protons from this region of the mitochondria to this region. Now this region is mitochondrial matrix, and this region is the intramembrane space of the intramembrane space of mitochondria. You need to go back and look into the structure of mitochondria. There we have talked about outer membrane, inner membrane, what is the intermembrane space, and all these things we have already discussed. So here this region is the matrix of the mitochondria. Here are some protons. So the energy which is released, when these electrons flow through these proteins, the energy which is released is used to pump the protons from this region to this region. To pumping means active transport, when these protons move from low concentration to the higher concentration. So here is a lower concentration of protons, here is the higher concentration of proton, but the energy which is coming from the high energy electrons is used to pump the protons from this region to this region, from this region to this region. Okay. And as a result of that, when there is an accumulation of protons in this region and there are less protons on this region, there is the formation of gradient. It's called electron-proton gradient. There are more protons on this side, there are less protons on this side. It results in the formation of electron-proton gradient, and then these protons can diffuse because there are more protons on this side, there are less protons on this side. They can diffuse, they can flow from high concentration to low concentration, and then they flow through another protein called ATP synthase, which is actually an enzyme. These protons, they move from this side to this side, from high concentration to low concentration through this enzyme, and when these protons flow through this enzyme, they activate this enzyme. And this enzyme is ATP synthase, and it results in the formation of the production of lot of molecules of ATP. And this process by which ATP is produced, this is called chemiosmosis. Okay, so this overall process in which the electrons are flowing through electron transport chain, and the electrons, they are pumping protons on this side, and then these protons are flowing from through this ATP synthase. This part is called chemiosmosis. This is electron transport chain, this is chemiosmosis. This overall process is called oxidative phosphorylation. This overall process is called oxidative phosphorylation. And in this process, energy is produced. A lot of molecules of ATP are produced. And in total, there are around 32 molecules of ATP are produced from 1 molecule of glucose. So some book says 30, some book says 32. There are some discrepancies of how the ATP molecules they are calculated. So this is overall process of cellular respiration. So this is glycolysis. In glycolysis, glucose is converted into 2 pyruvates, and in this process 2 molecules of ATP are produced. Then there is a conversion step. In conversion step, there is no ATP is produced. Then it's a citric acid cycle. In citric acid cycle, there are 2 molecules of ATP are produced. And in the oxidative phosphorylation, which we just talked about, which involves electron transport chain and chemiosmosis, there's a production of around 26 or 28 molecules of ATP are produced. And in total, there are 30 or 32 molecules of ATP are produced. And at every step, you can see there's a stripping of electrons, and these electrons, they are being carried by electron carriers. And where they are being carried? They carry to the inner membrane of mitochondria. So this is mitochondria. So this is the outer membrane of mitochondria. This is the inner membrane of mitochondria. In between, this is intramembrane space, and this is mitochondrial matrix. So 2 molecules of NADH are formed here, glycolysis. 2 molecules of NADH are in the conversion step, and then there are around 6 molecules of NADH produced in Krebs cycle and 2 molecules of FADH2. Six NADH2 and two molecules of FADH2. 6 NADH, 6 NADH and 2 FADH2. So this is overall showing the process of cellular respiration. Okay, I think I'm going to stop here, and the second part I'm going to record in the next lecture.